All right, so let's talk about meiosis now in the parts, and we're going to do this a little bit faster. So what I do want you to know is that meiosis occurs in humans, in males in the testes, and females in the ovaries. There are specialized cells that go through these processes. And so again, our diploid number is 46. So the whole purpose is to take one of these cells, and we need to get rid of some of the chromosomes. And because when you have an egg and sperm come together, if they are 46, then all of a sudden you have an individual with 92 chromosomes, which is not what you want. What you need is to reduce that. So this is actually sometimes called reduction division. So in humans, we're trying to get from 46 to 23. And to do that, we go through a process very similar to mitosis. But what you want to know is we actually go through it twice. And so that's what we're going to be doing today. That's what we're going to be showing you. All right. So as we go through, we'll start with meiosis 1, prophase 1. So it goes through an interphase, which your book doesn't really talk about well. So there actually is a doubling of chromosomes at the beginning. So what I want you to know, in humans, we would have 46 chromosomes. DNA replication occurs, and then we would end up with 46 chromosomes still. But now each chromosome is made up of 2 sister chromatids. So although we have the same number of chromosomes, we have double the DNA. All right, so then we go into meiosis 1, prophase 1, and something a little bit different happens. So you have your chromosomes with chromatids. Now in mitosis, we have something called homologous pairs. And what that is a like chromosome number 1, like the karyotype, where we kind of layer things together, look for chromosome 15 and put it next to the other chromosome 15. In meiosis, they search each other out. And so what happens is you end up with them pairing. And so this is where it becomes difficult because we have lots and lots of terms. So the homologous pairs find each other and they will attach by what's called a chiasmata, and the process is called synapsis. And so when that happens, these things will have other names, a bivalent, and they call it a bivalent because it's made up of 2 chromosomes, or sometimes they call it a tetrad. And that's often what I see. I think probably more likely today is a tetrad, which is basically 4 chromatids. So all of those are the same. A homologous pair that's combined, that's gone through synapsis, a bivalent, and a tetrad technically are all the same thing. So what happens here is something called crossing over. And crossing over is to increase genetic diversity. And we'll talk about that more on the next slide. And it happens at the synapse, or what we call the chiasmata. And it looks like something got unfinished here at the bottom. Yeah. So microtubules attached to the kinetochores, and actually for prophase 1, pull onto, I'm trying to think of what's necessary for you to know from right there. But those kinetochores, the microtubules, are basically trying to pull them kind of into position. So that's how they are being pulled together. All right, the next one is crossing over. And here's where the cohesion was talking about, where they're actually stuck together. And I guess I should have put that earlier too. But what you're ending up with, and what they're just trying to show you, is it kind of condenses. So here what they're trying to show you is 2 sister chromatids, okay? And then what happens is you got this crossing over is occurring between the structures. And the reason this is happening is genetic diversity, because you can see one's blue and one's red. And when the crossing over will occur, you're going to get this will be all blue, and then this little piece will be red, and this will be all red, and then this little piece down here would be blue. And what that is doing is mixing the male and female parts of your paternal chromosomes is really what it's doing. So it's called crossing over. And by doing this, what it ends up happening is when you get to the final is all four of those, what you're going to notice, the daughter cells, all four of them are different. Not only are they haploid, but even the chromosome that's in there is a mixture of, your parental information, the male and the female. So this works out really, really well. So again, crossing over, it only happens in prophase 1 of metaphase 1, and it is for genetic diversity. Okay, so this process only happens in prophase 1. All right, metaphase 1. Here, what you're looking at now is you have metaphase a little bit different. Before it was the all the chromosomes lined up on the metaphase plate, and really that centromere was kind of sitting on the plate. This time is a little bit different. It's the homologous pairs, bivalents, tetrads, however you want to call them, are now arranged on the plate. And so really what's sitting on there now is kind of the chiasmata. And so here what you get is independent assortment. And what they're trying to say here is, if you look at this picture, see how we have a red up here and a blue here. And then down here we have the blue and the red up on top. And then this is opposite. We have the blue on top and the red on the bottom. Well, independent assortment means how this one lines up has no effect on how this one lines up. And so that's what they're trying to show you here with possibilities. So if you look at if possibility 1 is blues are on the left, reds are on the right. And so what happens is you get babies that are blues, all blue and all red. But then if you look at possibility 2, a blue and red set up differently than the one on the bottom. So you end up with blues and reds in different combinations for different chromosomes in the egg and or sperm. Doesn't say if it's male or female. And so again, this is the equivalence, or not the equivalence, but the a way to introduce genetic diversity is, and they found, so no matter every set of chromosomes or homologous pairs you have, they're not affected by any of the other ones on how they line up, which one's on top, which one's on the bottom. So that is important. All right, then we get to anaphase 1, and the homologous pairs or bivalent tetrads are now pulled to the opposite poles. So instead of the sister chromatids being pulled apart like in mitosis, in anaphase 1 of meiosis 1, it is the homologous pairs, bivalents, or tetrads that are being pulled apart. And then in telophase 1 in cytokinesis, each cell has a complete haploid set of chromosomes. Okay, so in theory, in us, we went from 46 chromosomes to 23 chromosomes, and you're saying, why don't we just stop? We have what we need. But the problem is we have too much DNA. Each chromosome is still made up of 2 sister chromatids. So cytokinesis occurs, cleavage furrow, or cell plate, depending on animal or plant. All those things happen in the nucleus, just like before. Then we're going to go into meiosis 2. And there is a time briefly where it just sits. It's called interkinesis, between divisions. And what I want you to know is, although sometimes it's called a false interphase, under the microscope it might look like interphase, but no DNA replication. You do not want to add more genetic material. We have too much. So then what happens, you go through mitosis 2, which I'm not going to really do. It's the same as meiosis or mitosis, just with half the number of chromosomes, because the cell is now haploid. So here's what I need you to know. And I did not type it out. Okay, so let me make sure you do this. You start with 46 chromosomes in humans. Okay, and you have 92 chromatids. After meiosis 1, so and that cell is diploid. Okay, after meiosis 1, you end up with 2 cells that are haploid, which is what you want, except they have too much DNA because you end up with 23 chromosomes, but you have 46 chromatids in those cells for humans. So what has to happen is meiosis 2, which goes through the division again, no DNA replication. So you end up with 4 cells that are haploid and with single chromosomes where you want to be. Okay, so again, through meiosis, you start with a diploid cell. Through meiosis 1, you end up with 2 haploid cells, but with too much DNA, with too many chromatids. And then when you end up at meiosis 2 at the end, you end up with 4 haploid cells, all with the correct amount of DNA. And then to get back to the 46 is when egg and sperm come together. So this is kind of a comparison, and I would suggest you know this, mitosis to meiosis. And they're really comparing it, kind of meiosis 1 for the most part, and then you have meiosis 2. So know those differences, and that is kind of spelled out in this summary sheet here. And so last thing on this is sexual sources of variation. We've already talked about 2, crossing over and independent assortment. Crossing over, again, happens in prophase 1. Independent assortment happens in metaphase 1. Random fertilization. This does not mean that males just walk around and randomly fertilize females. That would be called PRISM. What this means is random fertilization is, millions of sperm are produced, and in females, multiple eggs will develop. Only one typically is ovulated, and so that would be the random fertilization. What egg is released, and what sperm actually gets there and ends up doing the fertilization. And then also mutations do occur, and those mutations then may or may not be passed on. So these are the 4 sources of sexual variation. And with that, we'll stop the share and recording.